Hello, everyone, and welcome to today's exclusive webinar focused on the critical freeze and thaw processes in the pharma and biopharma industries. So we are excited to have you join us as we explore the challenges and advancements in the vital area of drug substance manufacturing. And today we'll be discussing the importance of improving freeze and thaw processes to protect quality, enhance yield, and reduce the manufacturing footprint. So our goal today is to provide insights into the challenges and present a reliable and efficient solution that addresses them. I'd like to take a moment to thank our event sponsor, FARRAR. FARRAR is a brand of trained technologies, specializes in providing solutions for low and ultra-low temperature processes that are crucial for the manufacturing, freezing, and storage of medicines, vaccines, and other life science products. Their rich in and walk-in offerings cater to various critical stages along the biopharma value chains. And Trane Technologies is a global leader in climate control solutions, offering a comprehensive ecosystem of support for life science customers. They leverage the expertise of Trane, Thermo King, and FARRAR to deliver a complete portfolio of cold chain capabilities. This includes scalable solutions and an unmatched fleet of trained service technicians, ensuring effective temperature control ranging from plus 40 degrees Celsius to minus 86 degrees Celsius. So thank you, FARRAR, for your support in making today's webinar possible. And now it is my pleasure to introduce our esteemed speaker, Jeffrey Johnson. Jeffrey is the president of Biotech Design, a bioprocess engineering consultancy with a wealth of experience, 35 plus years in the biotech. He previously held key roles at Merck, where he was the new technology lead and director of global engineering solutions. Jeffrey was instrumental in assessing new manufacturing technologies, like for vaccine, biologics, and sterile manufacturing processes. He also led single-use technology initiatives, including CryoWall, DS container system. And now, without further ado, let's get started. Jeffrey, the floor is yours.
Great. Thank you very much, John. We'll start with today's agenda. We'll start out with an overview of how things are currently frozen in the industry and the challenges around freezing bulk drug substance. We'll look at the traditional ways this is done, as well as a newer alternative freezing technology that's offered by FARRAR. So a little background, little color. We always hear minus 80, right? We freeze our drug substance to minus 80 degrees. Where does minus 80 come from? Is it scientific? Not really. Minus 80 comes because dry ice sublimes at minus 79. And in the early days, things were held with large coolers filled with dry ice, which was at minus 79. So that's the reason why we have minus 80. It's not around your product. I mean, typically, liquid formulations of monoclonals have a glass transition temperature of around minus 28 to minus 31. If you do add sugars for lyophilized formulations, typically I've seen minus 40 to minus 44. And if you hold the product below glass transition, it's usually stable, right? That's where you want to be, below TG. And so this minus 80 really is a legacy of dry ice and not related to your formulation. Just a little background on why we hear this minus 80. I was always curious where that number came from. So the freeze technologies that are out there. On your left, and these are roughly to scale, so you can see we tried to make this to scale, the difference in sizes between these machines and technologies. On your left is an ultra low temperature freezer. I often call this an upright freezer. They're roughly the size of a large kitchen refrigerator, and they're very common, right? You see them all out. You see them, and it's very common in process development labs as well as in commercial manufacturing. In the middle, you see a plate freezer, substantially bigger machine. You can see this is a horizontal plate where you can see the plates, the shelves in the middle, where you place your containers of drug substance. Now here, this is set up and typically used for bags, 5 liter, 10 liter, 16 liter bags of drug substance in a shell. You place the shelves onto these shelves, and then the shelves collapse around it, hold them, and you freeze. And then on the right, you have the FARRAR controlled rate freeze thaw chamber. And this is a forced air convection blast freezer. So sometimes you'll hear me call this a blast freezer, and the one on the left, I'll call it upright freezer. The one in the middle is a plate freezer. So these are the traditional ways. And it's interesting, historically, the uprights have been used very frequently. The plate freezers seem to be more common in Europe, and I'm not sure why, but it's a more common technology offered in the European manufacturing scene. And the blast freezing is, for some reason, more common in the US. But again, people use all three technologies everywhere around the world. So we'll go through the pros and cons of these technologies and look at some detailed technical analysis of heat transfer, as well as some pros and cons of all three approaches. Let's start with the upright, the ultra-low temperature. Again, widely used in the industry for freezing drug substance, but that's not what these machines were designed for. They are a lot like a very large cooler with dry ice in them. They are designed for holding frozen material that's already frozen or adding a few samples. Very common to hold samples in analytical labs or small volumes. They are not designed around freezing of large volumes, 100 liters of bulk drug substance. And I'll get into this and show a technical analysis of why that is so. So let's look at that technical analysis. Some background, some theory here. When you're doing freezing, the chamber is around -80. The outside air is around 25 degrees, 20, 25. So you have around 100-degree delta T between your outside ambient room and inside that chamber. Because you have this large difference in temperature, there is heat transfer, right? You're going to lose the cold. The heat from the outside is going to be transferred inside, and it takes energy. It takes a compressor. It takes a refrigeration system to keep the chamber cold. Now, how is that heat transferred? There are three mechanisms. You have outside convection, the air transfer from the air to the walls of the freezer. Then you have conduction through the insulation of the chamber. And then you have the inside convection from the cold wall of the freezer to the -80 air inside. And when you look at this, the insulation is very good in these freezers, obviously, to keep it at -80. And it is an order of magnitude larger resistance. And so I'll really be focusing in this analysis around that conduction term. And so again, the top, we summarize this, right? We have the resistances, we have the convection, the cabin insulation for conduction, and the inside convection across this 100-degree delta T. And we're only going to look in this analysis at the conduction, like I said. So the equation for this, for conduction, is Q prime, which is the heat transfer, is equal to the heat transfer coefficient times the area for the transfer times that delta T divided by the thickness of your insulation. And so I have the constants below. You do the calculations around the area of a typical upright. And this also applies to the FARRAR chamber, because the FARRAR chamber is basically the same size as a traditional large ULT. And you come up with around 100 watts, 104 watts of heat transfer to keep that chamber at -80. There is also heat being transferred across the door. I won't go through all the calcs, but that's around 30 watts. And then you do lose some heat around the door gasket, around 24 watts. You add this all up and you get 158 watts. And so that's, 100 watts is, an old-fashioned large incandescent light bulb, right? That's the amount of energy you need. That's the amount of energy being transferred from the outside into the chamber. And so to maintain the -80, you need to have a compressor system capable of handling this 158 watt heat load. So if we look at a typical upright ULT and its design, it is typically designed at 65 to 75% duty cycle. And so what that means is you take that 158 watts and you divide by 0.65, 0.75, depending on the supplier, to give it a little bit of a buffer. And that gives you 210 to 243 watts. And so if you take the 210, the 243, subtract out the 158 that you need just to maintain the delta T, and it has an extra capacity, a buffer of 52 to 85 watts. And so that 85 watts is available to handle things like opening and shutting the door, or putting in a liquid at room temperature and bringing that, or 2 to 8, whatever, putting in liquid and cooling it down to minus 80. Okay, I'll use the 85 watts, which is the amount available at the high end of the range in my calculations. Now, if you look at the FARRAR, completely different machine, right? This rate chamber is an industrial commercial machine designed around freezing. It is designed to take liquid and freeze it and take out that heat. So again, the heat load across the chamber is the same. It's the same size. It's 158 watts. In addition, because this is forced air, it's a blast freezer, there is a large fan. That fan is putting in energy, and that's a lot. It's 325 watts. And so to keep the FARRAR cold, this blast freezer cold, you do need a bigger compressor system. 483 watts, 500 watts is required in this case to keep the chamber at minus 80 versus the 25 degree ambient. Now, if you take the 2100 minus the 500, you come up with 1617 watts remaining as a buffer, as compared to only 85 watts as a buffer in a standard ULT, over 18 times the amount of cooling capacity, refrigeration capacity in this machine. So let's put that into perspective. What does that mean? And so to look at this, I will use a 10-liter carboy as an example. Now, there are many different types and sizes of drug substance containers, right? There's 2-liter, 5-liter, 10-liter carboys, 5-liter, 10-liter, 16-liter bags, cryovaults, 16, well, from 1-2 liters, 16 liters, and 75 liters working volume. There are many containers out there, but a 10-liter carboy is common. I know one very large biopharmaceutical company that freezes all their drug substance in 10-liter carboys. So we'll use this as an example. So what we will do is we will look at the heat transfer, how much heat is being transferred at the moment when the liquid is brought down to 0 and the chamber is at minus 80. So this is the first moment when you're starting to freeze. And the big difference between an upright ULT and a blast freezer is the ULT, the air is static. It's called free convection. There is no forced circulation of the air inside the chamber, as opposed to the rate chamber, the blast freezer, where you are blowing air across it. And so the heat transfer coefficient is dramatically higher, somewhere between 2 and 10 times higher. And in this analysis, I will be conservative and only use a factor of 2 as the difference between them. And so if you look in the bottom right, I show all the coefficients used, 10 versus 20. You do the calculation. The equation here, is heat transfer Q is equal to U, that heat transfer coefficient, times the area of the outside of this bottle, times the delta T. And if you do that for a 10-liter carboy filled with 8 liters, you come up with 100 watts is the heat transfer. So you put this carboy into the freezer, and at the point when it starts to freeze, it can transfer 100 watts of energy by that convection. If you put it into the forced air, the blast freezer, because the heat transfer coefficient is twice, you need to be able to take out 200 watts of energy to freeze it at the maximum rate. So to put that into perspective, remember the ULT had 85 watts of duty, of excess capacity, but you need 100. If you want to freeze this as quick as possible, it is capable of transferring 100. You only have 85. So you're only at 85% of what's required for one carboy. Now the rate chamber, it has that 1600 watts, or it has the ability, that means, to remove the heat of 8 carboys, and it will do it twice as quickly. And so in the case of the upright, if you put more than one carboy in there, not only will it freeze more slowly, the compressors can't keep, the chamber temperature will come up will be warmer than -80, and it will take significantly longer to freeze. And so here is the theory behind it, right? This is why an industrial commercial machine designed around freezing is a better solution for quickly freezing your drug substance. 8 carboys versus 1 in this example. And so what? It's slower. Well, it's more than just being slower, although we'll see, with cryoconcentration, slow is not ideal. You want it to be quick, but it's more than just the time. The standard.
Jeffrey, if I can for a moment, so we have a couple of questions. So I will check with our questions moderator Rebecca from FARRAR, and we'll see if there are a couple of questions that she would like to address right now.
Yes, so apologies. I didn't want to interrupt your flow, Jeff, but we do have a couple questions.
That's great. Interrupt at any time, Rebecca. Feel free to jump in. Okay.
Perfect. So our first question is, what are the key parameters that influence the freeze-thaw cycle for drug substances?
It's the heat transfer rate. So it's the heat transfer coefficient, the area, and the delta T. So again, the more area you have, the more, and a key one is increasing this U, right? The overall heat transfer coefficient. And then the third one is the chamber temperature and the surface area of your container. Those are the 3 critical parameters.
Perfect. We do have a second question. How can the freezing rate be controlled to prevent product degradation?
So to do it as quickly as possible, you want to maximize your area and maximize your heat transfer coefficient. There are times when you want to slow it down. Say that your commercial container is 10 liters and you're in process development or clinical and you use a 2 liter, you may want to slow that down so that the clinical freezes at the same rate as the larger, so that you don't have, it looks really good in the clinic and then you find out when you scale it up to commercial that there are some issues because when you go bigger, it will be slower. And so the programmable machines, as we'll get to later, you can program the rate to slow it down to match between scales. The upright, it has no controls, it just freezes. And the small, clinical, preclinical samples will freeze quite quickly. And then you get to commercial and it can take days to freeze, as we'll see later.
Great. And we have one more question. What are the best practices for scaling up freeze-thaw processes from lab scale to industrial scale?
That's a great question. As you change the scale of your drug substance container, the freeze rate can change. And so there are machines out there, the Sartorius S3, I believe, and Meissner, they do make benchtop devices designed around matching the small scale PD scale, 30 mils, 100 mil samples, and you can program it so that it freezes at the same rate as 5 liters, 10 liters, 16 liters, or 75 liters, the scale that you want to use commercial, whatever your commercial platform is. And so I think that is the best practice, using one of these benchtop machines in your PD lab and actually slowing down the small scale process so that you get the same result that you will see commercially when you launch your product.
Great. Thank you, Jeff. That's all the questions we have for right now.
Okay, great. Thanks for asking and keep them coming. This makes it fun for me. So please keep asking questions. And Rebecca, Jan, please feel free to keep interrupting me as we go so that I can answer the questions. So back to the question, it's slow. Okay, let's say you have a very stable product. Cryoconcentration is an issue. It takes 3 or 4 days to freeze. That matches your manufacturing cycle. You can handle it being slow. Well, it's more than that, right? These machines, these typical upright ULT freezers offered by all the major manufacturers, were not designed for large volume, 100-liter scale drug substance freezing. And so what happens is it gives out this heat load that keeps the compressor running for days. And these compressors really are not designed to be run for days. They're designed to run for a little bit, bring it back down, take care of that 100-watt heat transfer, 158-watt heat transfer, just of the normal loss through the walls. And when you run these compressors all the time, because they're not designed for it, something called oil logging happens. And when you get oil logging, now you don't get enough refrigerant to the compressor, and the compressor fails. And so these uprights are they're reliable machines if you use them for storing frozen material as they're designed. They'll last 10 years plus. But we typically see, when used for freezing, that they can fail quickly, on the order of a couple years. And if you load them up with 100 liters, they can even fail quicker than that. And so you need to have a lot of redundant freezers, because you have to plan around the failure of these machines. So there's another commercial machine out there, widely used, especially in Europe, but it is again used in the US, especially the Sartorius Celsius system uses a type of plate freezer. One clear advantage, it does have a higher heat transfer rate. The heat transfer between the plate and the surface is around twice that of convection, air convection. So it's not twice, because you start to get some other resistances through the ice, but the heat transfer coefficient is around twice. It is very consistent. It's very repeatable. You have all these shelves, good contact, so it does freeze reliably and consistently. And because it is faster, it does reduce ice crystal formation, smaller crystals, and it reduces cryo concentration. So again, technically a very good solution. But there are some cons. One is these machines, they're complex, they're big, they're high capital costs. The machine shown here is on the order of close to $800,000, while the FARRAR 4000 series rate chamber that we showed was around $160,000. So again, around a 4 times difference in capital. And also because they are just more complex. These shelves have refrigerant flowing through the shelves, so the shelves are quite complicated. They have paths for the liquid to go through, and these shelves move up and down. So moving the shelves up and down at cold temperatures is mechanically complex, and you have hoses, and these hoses are connected through fittings, and these fittings leak. We are aware of one very large scale biomanufacturer that has a fleet of these, and they have a very large maintenance department and a very large maintenance budget set up just around the maintaining of these machines and handling all the leaks. So that is a con. The other thing is they're really only set up for single-use bags, right? It's a flat heat transfer surface. So for carboys, round bottles won't work at all. You'll have very little heat transfer surface. Cryovaults, the horizontal won't work. You need vertical plates. And so again, they're very well suited for using single-use bags put into a shell, but for the other images used, carboys, cryovaults, not very well suited. And the last con to them is there is longer labor time to set them up, load your containers, and unload the containers. Again, you have to load the shelf, and then the top shelf, these machines are quite high. It's like 8 foot high. And so to load that top shelf, you have to have a step stool, or often use mechanical assist devices to help you load them. So again, that adds some complexity and some time. So let's take a look at the FARRAR solution.
So Jeff, before we jump into the next section,
please.
We do have a question, and I think this is in reference to the earlier discussion about -80. The question is, what if the temperature falls below that -80 degree temperature set point?
And so below can mean either colder or warmer. It's not clear. If it goes colder than -80, which is unlikely, it's fine, nothing happens. If it goes warmer, until you get up to the glass transition temperature, it probably won't have a scientific effect. It will be a deviation. All these chambers, especially long-term storage, have a thermocouple that goes up to your BAS or some type of automation system. And if it goes out of an alarm point, you will get an alarm and a deviation to follow up. But if that temperature is colder or lower than numerically, the TG, scientifically, you should be okay, but it will be a deviation.
Great, and I
And
think
you often see that in the uprights, the ULTs, because they don't have a lot of capacity. If someone opens the door and doesn't shut the door all the way, or leaves it open longer than expected, you will see chamber temperature variations, and that can trigger off alarms and deviations.
Great. So that's all the questions we have for right now. And just for the participants, know that we are monitoring the questions. And if we don't get to your question right away, we'll either address it in the presentation or we'll hold it till the end of today's webinar. Thanks, Jeff.
Yep, sure. And so now I'm going to show some data to show that this blast freezer, for those that are traditional approaches, are either ULTs or plates. I'll show some data to show that these blast freezers do work quite well in an industrial commercial setting. So here we have some data provided by Integris around the freezing of bottles. So the first column is 90 2-liter bottles set into a FARRAR 4000. So 180 liters capacity of liquid being frozen. The middle is 4-liter carboys, and the one on the right is 10 liters of carboys with 112 liters total capacity. And so the average time to bring it from 20 degrees ambient down to 0, and then it stays at 0, as we'll see later, until it's completely frozen. And so the time shown here is from 20 until completely frozen in the solid phase, is on the order of 4 to 7 hours, with a fairly tight range of plus or minus an hour or two around that. And then once it gets to 0, you have to bring it down to minus 60. That's where the data was taken. And as I said, once you get below minus 50, you could actually unload your freezer there. So the time to take it down to minus 60 is a couple more hours. And so if we look at the total freeze time at the bottom, you can see it ranges from 5.5 hours for the small 86-liter load, which is less load and has more room around the bottles for convection, to 9 hours total. And the range is between 7 and 11 hours. So it's showing that up to 180 liters of bulk drug substance per load, freezing it in less than 12 hours, which is much less than 2 shifts. And so again, I think often in a commercial setting around shifts, and if you can get it done in less than 3 shifts, that means you get it done on day shift, you come in the next morning and it's ready, right? 2 shifts there. So all of this works. End of the first shift, you have it, you hit start, you come in the morning, and everything is frozen. You can also use these machines for thawing. And so a typical way of thawing of drug substance, especially those that don't have cryoconcentration or sensitivity to it, is to just leave it on a shelf in a room. And I am very familiar with a very, very large volume monoclonal, where at the drug product site, they take 16-liter bags and just wheel them into a room and let them sit there for 2 or 3 days until they're thawed, completely uncontrolled. And that is the blue line on the graph on the right. And you can see where it's pretty much dead flat out through 21 hours, right? Taking more than a day. Now you can speed that up. You can put a fan in the room, right? And if you put a fan on it, it is significantly quicker, and it's on, it's less than 16 hours. And if you really want to do it in a hurry, you can take the labor of opening the shell, exposing it, and blowing a fan on it. And if you do that, you can get it down to less than around 9 hours. That's the red line in the data here. But again, that's a lot of labor, right? And some risk of a brittle minus 80 container being opened and handled, where you do add some risk of integrity failure. There's a much simpler, safer way to get that quick thaw time, and that's to put it into a blast freezer, right? So if you put it into the FARRAR, you can see the green line here, where it thaws, and it starts to thaw in around 8-9 hours and comes up to 10 degrees C in less than 10 hours. And you can see the green line has a steeper slope, and that's because, again, the heat transfer coefficient is just higher in the blast freezer than it is in a room with a small fan on it. So if we look at the freeze-thaw cycle, here we have data from 9 5-liter shells put into the FARRAR. These shells, again, provided by Integris. They're the blast freezer shells designed, with slots to allow airflow in a blast freezer. And again, this data shows it starting at a very warm temperature, so it was water. They started it at the end of the last thaw cycle. Typically, you would start around 2 to 8, right? You'd put it in their refrigerator or ambient, so that would knock a couple hours off the data shown here. But you can see that all of these containers are frozen within 18 hours here. And again, if you started at ambient, it'd be around 16 hours, 2 shifts. And if you want to thaw, again, the data starting at 18 hours, it comes up and it gets to 10 degrees within 2 shifts. So again, showing in a shell, which is, when you're in a shell, it's a little slower because it's insulated. Shells vary in the amount of insulation, but if you use one of these blast shells, as shown here, you can freeze and thaw in around 2 shifts. So a little bit more detail around this blast freezer, how a blast freezer works. And so this is a picture of the FARRAR 4000 series. You can see it is a very high output cascade refrigeration system. It has a high stage compressor with 5 horsepower, 3.7 kilowatts. It has a 3.5 horse, 2.6 kilowatt low compressor stage. 60 air changes per minute. So you're really blasting the air through there, which at least twice, up to 10 times higher heat transfer rate. And it provides that uniformity, right? It blows around the containers. In a ULT, the container in the middle has a significantly different heat transfer than one close to the wall. The blast transfer, the airflow here ensures consistency across that chamber. And then again, around this mechanical robustness of these industrial machines, it has something called an electronic expansion valve. This is the piece of equipment not seen in ULTs, and it controls that oil logging issue. So oil logging is not an issue, and again, these machines are run for years and years and years with just routine preventative maintenance. The plus or minus 2 degrees, temperature consistency, and you can buy a machine with different automation options. A standard option is 3 preset freeze-thaw cycles, or you can buy it with a full PLC where you can customize and put in any thaw cycle or freeze cycle you like. Integrated hot gas heaters, so you can do this thaw cycle. What I have done is put in a 30-degree temperature at the initial part of the thaw, and then once it starts to thaw, you can cool it down to 20, and then after the 2 shifts, you can bring it down to 8 degrees if you want to, so that when you come in, you already have a cooling product. And again, you can program the machine anywhere between plus 40 down to minus 80. And so now on to some practical engineering facility summary of these machines. In the picture up front, we tried to show them to scale. This is to scale. On the left, we can see the plate freezer, a very large machine, expensive. It can hold 24 10-liter bags, so it does have a larger capacity. And it takes around 7 hours to freeze the 10-liter bags. In the middle, you can see the FARRAR 4000. On the right, a typical ULT. You can see the green versus the blue. The chamber sizes are quite similar. The rate chamber does have the black box on the side where you have the additional mechanicals and controls. And here, you can put in 10 10-liter bags, and you can freeze them in around 12 hours. On the right, the ULT, typically you'd only want to put 5-liter bags in there. It's 50-liter capacity. It takes 3 days to freeze that. You could theoretically put in 10 10-liter bags, but it would take significantly longer, right? Maybe 4 days to freeze. And so this is kind of our performance footprint summary of the different options. You can see that the capacity of the plate freezer is 2.5 times greater, but it is 4 times the price. And so you could buy 3 of the FARRAR freezers, get more capacity, lower capital costs, and have the redundancy of having 3 versus 1 of these machines. And so here's a summary of all the different attributes we've discussed and talked on one slide. If you want to hold already frozen bulk drug substance, the standard ULT is the right answer, right? The forced air convection and the plate freezers are not designed around long-term storage. And if you want, for some reason, like if you're in a PD lab or early clinical, you want to freeze and store in the same chamber, the ULT is the right solution because it's the one for long-term storage. But other than long-term storage, you can see the blast freezer and the plate freezers are the commercial industrial solution. They are designed for freezing. The uniformity throughout the chamber is much higher in both forced air and plate versus the ULT, where you see very large variations of the wall versus the center. The flexibility. Now, this is really important if you're in your preclinical, early clinical development where you have different size containers. You may have cryovaults for some products and 2-liter, 5-liter, 10-liter carboys or 5, 10, 16-liter bags. You can put any size in the forced air convection or the ULT. The plate freezer typically is only around where you have flat surfaces. Easy handling. Again, the forced air convection, the ULT is the same thing. You have shells and you load it. The plate freezer is much more complicated because you have to load and then move the plates, and it's a bigger machine and you have to set them up high over your head. The configurable freeze rate, we spoke earlier. The first two machines are programmable, although ULT, it's just on off. Again, integrated predictive preventative maintenance, as you see on commercial industrial machines, standard on the blast freezer and the plate freezer. A ULT, now it works till it breaks, and when it's broken, you get an alarm and you quickly run out there, move everything from the broken freezer into the spares that you have to have because you have to plan around these breaking. Controlled thaw, again, an upright has no thaw capability. Both the plate and the forced air can be used for thaw, and you can program it to program initially, if you want to, initially at a high temperature and then lower it later in the thaw cycle. Capital costs, the ULTs are by far the lowest capital cost. Then we have the blast freezer, and the plate freezer is by far the highest capital. Maintenance and reliability, again, both the forced air and the plate are good solutions. They are industrial. The plate freezer having the added complexity and the leaks around the fittings, and the ULTs, they are used. They're not red. They're used widely, but they do break down frequently, and you need to have lots of backup capacity. And then around the freeze rate, the plate freezer, the best, as we've seen, around 10, 12 hours for the forced air, so it is very quick, while the standard ULT, I couldn't call it red because it's getting widely used in the industry, but if you can take a 3-day freezing cycle, but it is significantly worse than the other two options. So I think the takeaway from this is use the right tool for the job, right? If you want to freeze, use a machine designed for freezing. And if you want flexibility of using different sizes or shapes of container, use the blast freezer as opposed to the plate. If you want to store your material, a ULT is a fine solution. There are also options for large volumes where you can get a walk-in, custom walk-in -80 freezer. But again, for storing, you want to use one of those solutions. So to go back to the introduction from Jan, FARRAR, I've known them for a very long time. They're now part of a much larger corporation, Train. And so with this, the support and backing of Train is able now to offer an end-to-end portfolio of cold chain capabilities, scalable solutions, and a fleet of technicians to maintain all of this equipment, whether it be in the freezing or storage or transport of your drug substance. And with that, I'd like to hand it over for Q&A.
Perfect. Thank you so much, Jeff, for that informative discussion. So before we kick off the Q&A here, I would actually like to introduce Scott Masella. He is a portfolio leader with FARRAR, who is joining us today for the Q&A. So welcome, Scott. We're going to be going through our question list, which thank you all for entering so many great questions into the Q&A box. So the first question is, what strategies can be used to reduce the impact of freezing on the stability of biologic drug substances?
I'm sorry, could you repeat that one?
Sure. What strategies can be used? Oh, it went away. Come back.
You can see it on top of the webinar window. So
Oh,
it is
thank you, Jan.
highlighted.
What strategies can be used to reduce the impact of freezing on the stability of biologic drug substances?
Well, again, your formulation has a lot to do with it. So when I started out in the 90s, the formulation of monoclonals were not as well known. Cryo concentration was more of an issue, but I think the best solution is to get a formulation that works for your molecule, so the cryo concentration is really not an issue. The other one is freeze it quickly, right? So get it down under 2 shifts. And if you have a particularly labile molecule, you may want to go to other technologies such as liquid nitrogen, right? I'm very familiar with live viral vaccines, extremely sensitive to temperature, and liquid nitrogen blast freezing is a common technology in that case. But again, for modern monoclonals, either the blast freezer or the plate freezer usually gives you a very good quality end result.
Great. Thank you, Jeff. So our next question, what are the effects of repeated freeze-thaw cycles on drug substance quality?
Yeah, that's a great question. Again, I'm not a formulation scientist, so I'm not the best to answer this. But again, it comes back down to how good of a formulation do you have, how quickly do you freeze it, and how controlled is your thaw. If you're putting it out into a room and holding it for 2 or 3 days ambient, it's not going to be quite as, and repeat that over and over again, it will have some impact on product quality. And also, if you do it over and over again and you're using bags, you start to get into an integrity issue. You know, this repeated handling, taking it down to -80 makes them very brittle, up and down. You can start to see more leaks. So there, you might want to use a carboy or a cryovault. Again, you know, cryovaults being a much more mechanically robust solution than a 2-liter bag.
Perfect. All right, our next question. How can the environmental impact of freeze-thaw processes be minimized?
Yeah, that's a good question. If you can use -40, if you have liquid formulations or your TGs are warmer than -40, you can use other technologies, other refrigerants that have a different carbon impact, carbon footprint. So that's one solution, is freeze to -40 as opposed to -80, and use more environmentally friendly refrigerants. Scott, do you have any insight on that one?
You know, the only other thing that I would add is you kind of touched on it at the end when you mentioned -40 or -80. So in order to get the amount of cooling capacity, in order to freeze drug substances, bulk drug substances down in a fairly quick time frame, you have to use a second stage refrigerant that has a pretty high GWP. Using refrigerants like hydrocarbons, the volume of refrigerant that you would need is too high for safety standards. Certainly in the US and in the EU, the safety standards you have to have for your electrical equipment would not allow the volume of gas needed as a refrigerant. So if you can, the impact of going warmer both helps your energy consumption, but it also helps you to be able to use something other than a high GWP refrigerant.
Great. So our next question is, how do you ensure consistency in freeze-thaw processes across different manufacturing sites?
Well, the best answer to this is put in the same technology at all your sites, right? So fill into the same container and put it into the same freezer and freeze it the same way at every site with the same load pattern, right? So if you are an older company using uprights, make sure you maintain the same load pattern. Because if you put just 20 bottles in versus 80 bottles in, you're going to see a very significant difference in your freezing rates. With the plate freezer or the blast freezer, because it's much better controlled, you will see much better consistency between sites. With the blast freezer, again, if you have the same cooling capacity and the same cross flow rate, you should get similar heat transfer coefficients, and you should see consistency across sites as long as you do the calculations in the test to show you're getting the same velocity and temperature profile. But the best solution is just buy the same technology, put it in the same machines at all your sites.
Yeah, nothing to add there. That's good.
And again,
right.
if you do have multiple images, right, and some sites have bags and some products have bottles, and you're using a plate for the bag, and now you have a product that comes up in a carboy, you'll have to buy a different technology, right, like a blast freezer for freezing that bottle. You don't want to be putting round bottles into a plate freezer.
All right, our next question is, can samples be stored in the blast freezer, and how often does it need to be defrosted?
I could take that one, Jeff, to start.
Yeah.
So yes, you can store samples in the blast freezer for a short period of time. We've done those studies. If you don't open the door, you don't need to defrost it. And we've run the studies to go as long as 4 weeks. But our recommended use case is run your freezing cycle. When it's complete, you could wait a day or two. It depends on on your process. Remove the material, put it in a storage freezer, and then run your defrost cycle. So the defrost cycle on the blast freezer, unlike our upright ULC series, is a manual defrost. And by manual, I mean you manually engage the defrost function. You don't want to do that because it only has one refrigeration system. You don't want to do that with samples in the unit. But once you finish your run, you run the defrost cycle, and then the unit is ready to be used for the next application. But again, we run the studies, and we can continue to do a PQ type study like that if anyone is interested, to go as long as about 4 weeks without opening the door.
Right. And so again, well suited for small scale startup. You could load it up, run a freeze, program it to hold it at -40, -20, -30, whatever your long-term storage temperature is. If you want to, hold it for a week until your next batch comes through, right? So if you want to hold it in, if you have a 2-day cycle, a 3-day cycle, 4-day cycle, 1-a-week cycle of the next batch coming through, you can set up your manufacturing around that cadence, but you don't want to hold it longer than a month.
All right, so our next question is, what utilities are required for each type of freezer that was presented?
I'll take a stab at starting, but I think the final answer will be, we'll probably want to get back to you to be for the exact details. But I'll start with the ULT. So the ULT requires either a standard electrical outlet that you might find. Typically, it's a 30 amp circuit. It could be 230 volts. Or if you're in North America, it could be 115 or 120 volts. No other requirements there. There are no water-cooled options. For our rate chamber from FARRAR, it is a 3-phase 60 hertz or 50 hertz application with air or water cooled. So if you have air cooled, then you just need to worry about the electrical requirements. If you have water cooled, it supports tower water or chilled water. And those rates are listed in our specification sheet that I don't have off the top of my head. For a plate freezer, it would be similar power requirements. as that FARRAR freezer, and it also has water as well for water cooling of its refrigerant system. And again, we can probably pull the exact details out and send those afterwards.
All right. Next question. What future trends in freeze-thaw technology are expected to shape the pharmaceutical industry?
That's a good question. There is some newer technology out there for handling larger volumes in your drug substance container. So up until recently, 16 liters was the largest you would see being frozen typically. There are now 2 different solutions out there that can freeze 75 liters. Meissner has the CryoVault, and Sartorius now has a 75-liter bag option, both using blast freezers, by the way, as the technology for freezing. That's the latest I've seen.
I think that I would agree, Jeff, like the trend of minimizing the failure points and taking a bit more risk with, if you have a failure, you definitely lose a larger volume of material, is the trend for especially what I would consider drugs that have large volumes associated with them, as far as things like vaccine, weight loss drugs, things like that address a large population. The other part is working on lowering the freezing point using pressure or other means to try to not have to store so far away from ambient, as far as delta T away from ambient. That's years away from being approved for use in general manufacturing, but that's those type of trends that we're working on in our own development areas. How can we lower the delta T so that we can get there?
And I'd say another trend is integrity and material handling and shipping of the drug substance. So again, 2-liter bags, any bag, there is an EVA, EVOH oxygen transfer barrier, which has a TG of like -30 something. So once you get below -40, you actually have a glass bag. You don't have a plastic bag, and they become brittle, and you get leaks. And integrity is a euphemism for leaks. So how do you prevent leaks? And one solution is CryoVault, which is just a more mechanically robust TG of -110. Better shells, right? So you can see shells that are more have better dampening foam. Insulation is to prevent integrity, but the more foam you put around it, the slower they freeze. So maybe coming up with and we've seen this, Integris offers shells that give you good heat transfer with blast freezing and give you good integrity. So that's a trend. The other one is getting material in and out of the freeze cycle, and how can we simplify that, get it onto a pallet, looking at the overall process of getting it from your bulk drug substance tank in the post-viral area until you get into the formulation tank filling your filling line. That's one process, right? How do we optimize that entire process from tank to tank? So that's freezing, shipping, logistics, and the thaw cycle.
All right, so our next question is, what tests are used for the verification of a freezer and ULT?
Temperature mapping, right? You validate temperature maps of the chamber. That's a well-known practice, and if you don't do it internally, there are lots of firms that can offer validation packages around validation of a freezer. Scott, anything to add there?
If you're going to use the unit for freezing studies, we've also mapped the ideal locations for the vessels that you're putting into the freezer, so to get repeatable results as well. So if there might be some mapping, I know we do it with computational fluid dynamics to start, and then that kind of gives us a guide towards how to do that process study. But then that's the other thing we'll do after mapping the mapping the unit, is that we'll do that study as well.
Right, and you probably want to do a more thorough study up front to qualify a machine, and then after you do that, you could buy multiple machines with a less rigorous validation.
Correct. Yes.
All right, our next question is, what is the maximum load of water-like product to stay within the capacity of the compressor in the FARRAR controlled rate chamber?
Right, so as we saw, 1600 watts and 200 watt heat transfer coefficient. So theoretically it's 8 liter containers, 64. However, that's if you want to do the optimum quickest possible time. But you can put significantly more in there. The chamber may not be at -80 the entire time. It may get slightly warmer, -70, -60 during the highest heat transfer load, but the compressor can take it fine. So unlike a ULT, where if you run the compressor longer, you risk failure, if you load this FARRAR up to its maximum capacity and pack it full of liquid, it will take longer to freeze, but it will still be mechanically robust and able to handle it. And similar with a plate.
Yeah. The only thing I would say is, as you were explaining it, Jeff, the one thing I wonder if there's some questions is when you talked about it may warm up to -60. So the air, when we set that blast, the FARRAR unit on blast, the unit is running at -80 and is moving air at 1 air change a second through the system. When you have that much material in there, the air is going on its return path is not going to be -80. It's going to be probably in -60 range, at least until the bottles get to -60, and then it'll start to cool down from there.
Right. Well, and also what happens, Scott, is the calculation I did is with no ice on the walls. So that is the maximum heat transfer rate. So once they start to freeze, you start to build up ice on the wall. That slows down the heat transfer rate. It's less than 200 watts, and then the machine will catch up.
Correct. Yes.
Right. So that 8 liter carboys is at the absolute, is the maximum heat transfer.
Great. Our next question is, how do you balance cost effectiveness with maintaining high quality freeze thaw processes?
Yeah, there's a great question. I guess the best way to do it is look at your portfolio of molecules and see where cryo concentration starts to be an issue. If it can take a 4-day thaw cycle or freeze cycle, maybe ULTs are an answer, as long as you're willing to have a fleet of them and lots of spare capacity for and handling the deviations around mechanical failures. For most modern formulations, less than 2-shift freeze gives you a quality product. If you do have a formulation or a molecule that's particularly labile, you could look at smaller containers with more surface to volume ratio to increase that heat transfer coefficient, right? Or even get into liquid nitrogen blast freezing, which is, again, a much more expensive approach, but it's extremely quick and you can do it in less than an hour. So again, it's the surface to volume ratio of your container and the heat transfer coefficient.
All right. Next question. How does energy consumption vary between the different freezing technologies? And what are the implications to sustainability?
Yeah, good question, Scott. I'll get you into the details, but if you look in the overall amount of energy used by these, it's in your overall plant, it's a relatively small number. If you were to do a Pareto chart, it would be way down on the Pareto chart of your energy consumption, and probably negligible compared to your air handlers in your room or even lighting in your room. But Scott, I'll let you get
the one thing I would say is that with both the plate and the FARRAR blast freezer, when you're not using them to freeze, they can sit idle. They don't need to be running. With your ULT, you're going to be running it all the time because you can't put product in it, pull it down, and start it from ambient like you can with the other two. So that's going to be that'll also vary your energy consumption as well. So while the plate and the blast freezer have significant refrigeration capacities that are much higher than a ULT, you're also not running them as often either.
The other difference is the plate freezer does not have the blower. Mm-hmm. So the blower does put energy in, and so the energy consumption of a blast freezer is higher than the plate freezer. But is it significant, and how big of a carbon impact is it?
Yeah, it's not very significant. Like the differences are pretty minimal.
Yeah.
Next question. After door opening and closure, how much time is taken for temperature recovery?
Boy, we have lots of data on that, Scott. Do you remember it? I don't have it in front of me. There's lots and lots of data.
We have lots of data.
Or at some point.
We have lots of data, but I guess the first thing I had about this one is with the you're usually not opening the door and closing it during a freezing process usually. So that doesn't really come into play with respect to the blast freezer or the plate freezer. With a standard ULT, we have oodles and oodles of data on that, and depending on how long you open the door, let's say 1 minute, you could have a recovery time that's in the 1 hour range to get back to your temperature. But again, during your freezing process, it's not often that you'll open the door. It's usually this is a measurement typically reserved for steady state operations.
Right. But during storage,
during storage
is when you may open up the door and grab a certain couple bottles. And you're saying it's on the order of an hour for recovery?
Yes. If you have a minute, yes.
Well, right, okay.
I think you'll see published reports in a lot of data sheets that might say like 8 minutes, 12 minutes, 22 minutes. But most often those are to get the unit back within the alarm threshold, but to actually get it back to its temperature, let's say at -80, it's going to take well over an hour.
Next question. How can you minimize the risk of cryoconcentration during the freezing process?
Did we already address this one?
We may have.
I think we did. Again, I'm not a formulation scientist. I have a lot of experience talking with formulation scientists in the development of the cryovault. Again, better formulations, right? Get a formulation that's ideally suited for your molecule, and to freeze it as quickly as you can.
Yeah.
All right, fast follow. What are the common challenges faced in the freeze thaw process, and how can they be addressed?
So, I think I spent a lot of the time designing, building, and running biotech factories, and focusing on drug substance freezing. Integrity is the biggest one, I think, which is why I said taking the shell and opening it is not a great solution, because you're opening a shell of a -80 bag, which is handling of you want to minimize all handling at -80 because they're brittle. Other solutions like cryovault are designed just to be more robust. Handling of it, how do you get it into the freezer, out of the freezer? How do you ship it? Distribution and logistics actually is a big deal. So shipping of the drug substance frozen from drug substance site to drug product site, which is often, across the ocean. And so you have to fly, and those containers and how you do that and the handling and validation of that is a big issue. So there are a lot of challenges. Thawing, how do you get it thawed? At what point do you thaw? How do you keep it from getting too warm? I had one process where we had to get it till it was like a slushy and transfer it at that particular stage. You couldn't thaw all the way. So hopefully you can get, like monoclonals are more stable. Typically you can bring them up to ambient. But again, handling of this product is a real challenge. The freezing of it, I'd say the big challenge there is loading and unloading of the freezer and the material handling around that. And the bigger the container, when you get up to these new 75-liter containers, obviously you're not going to pick these up, right? And you need mechanical assist for those. Scott, anything to add?
No, you got it.
All right, next question. What are the regulatory requirements for freeze-thaw processes in the pharmaceutical industry?
Maybe we should get back on this. I'm not a regulatory science. So again, you have to validate it, right? You'd have to do the mapping studies. You would have to file what your freeze-thaw process is.
Mm-hmm.
So once you file it, it's going to be very, very difficult to change. Again, it would be refiling. So again, it's part of your PPQ. So you'd have to qualify the equipment and validate the process.
And in fact, Jeff, we've seen this a number of times as customers would like to move away from ULTs. They find that, in let's say going to the blast freezer, they find that they would have to re-qualify because the blast freezer would be so much faster. So what they often do is that they'll take the blast freezer and they'll apply a control rate process to it to have it mimic the ULT. So it'll go as slow as your ULT to match the regulatory filing so that your process doesn't have to change. The only difference would be you save some ULTs, and your ULTs are now used for storage. So once it's complete, you move it to the storage freezer. Your ULT doesn't have as many service issues, but your process remains the same. And we've had others that said, you know what, we're redoing our validation. We're going to see how fast we can go so we can improve our processes because we're losing yields. Could be yield loss. It could be process. It's a long pole in the process tent. So we've done that too. But you're right. You have to match what you file.
Right. And again, I am aware of the thaw process being important. If you thaw it over multiple days, you can get some aggregation, which can lead to product quality impacts and filtration issues at the drug product site. And if you thaw it more quickly, like in a blast freezer or a plate freezer, you may see less aggregation and you may see improved product quality.
Great. So our next question, how can the latest advancements in freeze-thaw technology optimize the supply chain for biologic products, ensuring both cost efficiency and the preservation of drug efficacy during transportation and storage?
Yeah, this goes back to what I said earlier around the whole end-to-end solution, right? So and again, formulation scientists aren't focused on this. They're focused on freezing of my molecule, thawing of my molecule, without looking at that overall DNL from the tank in the drug substance post-viral area to the formulation tank filling your filling line, right? It's an overall supply chain. And so shipping, distribution, logistics, holding your drug product, your drug substance at -40, -30, -20, -80, whatever your validated temperature is for drug substance storage, all need to be optimized. I am aware of many companies that hold at -80 for drug substance storage. And as I showed, if your TG is -30 or -40, you don't need to do that, right? You can hold it at -40. By doing that, that allows you to use a much lower cost walk-in -40 freezer. And that has a much lower environmental impact, much lower capital cost, far more efficient. You can have racks, pallets, forklifts handling it. palletizing. How do you move this drug substance around? So your shipper, how many liters can you get in there? It's the material handling aspects of this that can make it more or less efficient. Again, I'm biased. I worked on CryoVault. I spent a lot of time on CryoVault. I think the DNL of CryoVault is the next generation. And if you're using 2-liter round bottles, you're going to see lots of complications in handling all those small bottles. So again, don't look just at the freezing of your container. Look across the entire DNL supply chain.
Okay, during scale-up batches or development batches, how will CQA be checked?
During which batches?
Scale up or development batches. So early drug development.
Right. Again, I've seen where we put thermocouples into containers and check the freezing profile. I've seen multiple thermocouples within a drug substance container. Sometimes you map 1 or 2 containers and with multiple thermocouples, put it into a large freezer filled with your validated load of 90 bottles or 10 bags, with a couple of them thoroughly mapped to show worst case or last place to freeze at scale. And then once you have that data, you can use some of these scale down systems like the Sartorius, I think it's the S3 and Meissner, so that you can do 30-liter and 100-liter bags, milliliter, right? 30 milliliter, 100 milliliter bags. Slow it down using a programmable, this benchtop is designed to slow it down so that you can freeze these small volumes at the same rate as the large volume commercially.
All right. How can you mitigate risks of a contamination during the freeze-thaw process?
Yeah, this is, again, closed systems is how you do it, right? And I am familiar with an extremely large volume biologic where they're still filling 2-liter carboys open in a hood on the manufacturing floor, hundreds per batch. And they have a biosafety cabinet and someone's in there manually filling open bottles, hundreds of them every batch. And this is a multi-billion-dollar product, still being done that way. But it's an older product. Modern products, you don't want to do that. I don't even know if the FDA they would certainly frown on that. The expectation in today's technology is for it to be closed. And there are multiple solutions, both carboys, bags, CryoVault. They all offer closed capabilities.
Jeff, is there any risk of any issues within the freezing process itself, like bags breaking or cracking that need to be considered as well?
Yeah, that's something I did think of wrapped up there. Is integrity is the issue, right? So if you do get an integrity leak, what do? And do you want to throw away the bag, which contains $100,000 worth of product? And the answer is probably not, right? It's not sterile at that point. You transfer it and do a bioburden and do an investigation and show that it was acceptable. But and that's a lot of work, right? So integrity is a big issue. And again, these are more robust solutions minimize those integrity. Or I like to say, if you're using bags, you can put a better Band-Aid on it by using a better shell to increase or minimize the risk of an integrity issue.
All right, next question. How can advancements in freeze-thaw technology improve the safety and consistency of biologic drugs during large-scale vaccine distribution?
Yeah, I mean, especially vaccines, the big issue is the last mile, is what they call it, getting it out there. And so, again, if you can get a formulation that's stable at -40, it's going to be a lot easier to transport than something that's at -80. It's the last mile that's a huge one in vaccines. And again, if you have a live viral vaccine, they are extremely temperature labile, and blast freezing or alcohol shell freezing or other technologies that we've discussed today might be required. But for typical therapeutic proteins, and especially monoclonals, they're fairly temperature stable, and the technology we discussed are typically fine.
Okay, we have a handful of questions left.
The other one is just think about those shipping. When you ship from your drug substance to your drug product site, I've worked with shippers that have a 5-day validation. I've worked with shippers that have a 7-day validation. They keep it, within temperature range over that period. That's actually a big difference, because getting it through, to the airport, onto the plane, off the plane, and through customs, 5 days can be a real challenge. 7 days gives you enough buffer that you can actually use a cheaper distribution method. The 5-day, often you have to have a person escorting it the whole time. 7 days, you may not need that. So that's another thing to look at is the validation of your shipper.
Mm-hmm. It's a good ad.
Okay. Next question, what quality control measures should be in place to monitor the freeze-thaw process?
Good question. I think we all have a thermocouple in the chamber, for sure. Most people don't have a thermocouple in a dummy bottle. You could theoretically think of having a bottle in there of water with a thermocouple to track that. Commercially, I'm not familiar with that one. It's more of a PD type activity.
That's a feature inside the FARRAR unit that you can have. There's a second probe that'll track on the display, so you can see it. You can track a sample bottle along the way.
Yeah, when I was using the FARRAR, we used to have one on the inlet, one on the outlet, but to show consistency. But on the plate freezer, again, I'm not familiar with them. They're more of an EU machine. I've not used one. Well, I've used the Celsius, but how you do it on that? I guess you'd measure refrigerant. Yeah, how do you do it on a plate? It is relevant.
I think it's I've only ever seen it where you've run your validation samples and you track knowing where it is based on the time and assume consistency.
Yeah, so you do it during your development, but not during commercial.
That's right.
Or maybe we'll get back to you on that one.
That's something we'll have to look into.
All right, we're down to our last two questions here. So how do you ensure uniformity in the freeze-thaw process across large batches from a QC point of view?
QC or QA, I would say QA. But again, typically what I've seen is these mapping studies done during qualification, where you map the chamber during operations, and you may have I've actually mapped every container in the freezer during development studies with water, not with product, to show, how. And when we showed the data here earlier in the presentation, showing that same thing, to show consistency across the chamber. Right, and then as long as you have the thermocouple in the chamber and you have your airflow, you do have consistency batch to batch.
Great.
Yep.
And to close out our Q&A for today, what advancements in cryogenic storage technologies are emerging to improve freeze-thaw cycles?
Scott, I'll let you handle that one.
So the advancements in the storage technologies are number one, I think that everybody would probably agree that the best way to store material is at these temperatures is probably in a chest type formation. As you get colder, the air in the chamber gets significantly heavier. So when you open an upright, even though the upright's very convenient to pull material in and out of, all the air falls out. which means that the recovery time is a lot slower and longer than if you were able to hold on to that cold air longer. So taking the advancements that we're working on is how do we take that recovery time that's in a chest freezer and put it into an upright formation? How do we reduce our need for, so that's the first one. So using forced air technology to do that. The second one that I would say that we're working on and the industry is working on is how do we reduce our dependency on consumables like liquid nitrogen and dry ice. So while they're not, I wouldn't say they're improving freeze-thaw cycles, they're maintaining the current freeze-thaw cycles while driving towards a net zero goal that the industry is looking for. So those are the top two that I would list. Focuses that FARRAR and others in the industry are focused on.
And I thought of one other while you're talking, Scott, on storage. Is the ULT uprights versus a more in the old days custom walk-in freezer.
Yeah.
And so I worked on a product that was -80, a very, very large volume. It was actually an intermediate product. And we had custom -80 walk-in freezers that turned out to be extremely robust, and after 35 years are still running fine. And that was US based. We went to Ireland. They said, oh, we don't have that here. We want uprights. So we put in 80 upright freezers. And then after a couple of years of maintaining them, they got frustrated and decided to go with the walk-in solution. So there are now more suppliers in the EU offering these. And again, FARRAR is now offering a more OEM off-the-shelf, larger volume. So I'd say one advancement in storage is larger volume off-the-rack OEM type solutions.
Yes, that's a good point.
All right, so that concludes our Q&A for today. So thank you to Scott and Jeff and everyone on the call for such a lively Q&A session.